Welcome back, everyone. So this is our second video for this unit's lab. So this particular second part will be actually working out of the lab manual. So if you have received your lab manual, then you can just work directly out of it. It's our first lab topic, one scientific investigation. And it goes from page 1 to 21. If you don't have your lab manual yet, you haven't, hopefully you've purchased it, but if you purchased it through the online bookstore and it hasn't arrived yet, we do have a copy of it here. So you can just follow this link and you can complete it with the PDF version, okay? So that's available, but do realize that we won't, I think it's after lab topic 2, we won't be providing links anymore. So you do need to purchase the actual lab manual, okay? So this particular lab manual, again, you just get from the bookstore. So that's the only required purchase that you need to make for this course, okay? So let's go ahead and take a look at our lab. So this is what it looks like when you open up your lab manual. So lab topic number 1, and it's a scientific investigation. So by the end of this lab, you should be able to identify and characterize questions that can be answered through scientific investigation. You should also be able to define and explain what characterizes a good scientific hypothesis. You should be able to identify and describe the components of a scientific experiment and develop hypotheses and predictions for a scientific investigation. And you should be able to design and conduct an experiment to test the hypothesis and summarize and present results in tables and graphs. And then finally, discuss results and critique experiments and interpret and communicate results. Okay? So we're going to go through the different exercises, and I'll just kind of, run through them with you, and then you can perform the information or experiments or whatever at home. Okay? So the first part, exercise 1.1, has you basically look at the first step of the scientific method. So questions and hypotheses. So in lab study A, it has you basically think about asking questions. So the first step of the scientific method is making observations, which helps you come up with some questions that you want to ask. Okay. So that's lab study A, and then I think there's some questions that are associated with that. And then the second part of exercise 1.1 is lab study B, so developing hypotheses. So remember with developing hypotheses must be testable and falsifiable. And so you're just going to follow the information, and then you're going to identify what's a good hypothesis and come up with your own. So in exercise 1.2, you're going to be looking at an experiment, and it's a particular case study investigating adult monarch butterfly. And in this case, you're going to try to figure out if they have a preference for food plants that reduce parasitic infection in their offspring. So we have basically parasitic infected and uninfected female butterflies mating in cages. And then the butterflies, they lay eggs in separate cages containing 2 milkweed plants with different levels of anti-parasite chemicals. So some of them, the AC, that indicates high levels of the anti-parasite chemicals, and then A1 indicates low levels of the anti-parasite chemicals. So basically, you're trying to figure out which plants the butterflies prefer to lay their eggs on. And then so you're counting the number of eggs that are laid, and then you repeat the experiment 2 days later. Okay? So based on this, experimental layout, this experimental design, you're going to state your hypothesis. Okay, so based on this information, what would be a good hypothesis that's not only testable, but also is falsifiable. Okay, so you'll just follow the information from the exercise and answer the questions along the way. Okay. Next, we have exercise 1.2, continuing on. So in designing experiments, we're going to be to test our hypothesis. We need to determine the variables that are involved. So we need to understand the difference between a dependent variable and an independent variable. So we talked about this in the previous lab video. So a dependent variable is the variable that will be measured and counted or observed. Okay, so think about what we're actually doing in the experiment. What are we counting? So think about like what's being measured in that particular experiment. The independent variable is what we're deciding ahead of time that will be manipulated or changed. Okay, so think about the treatments, the groups. What is the difference between the groups? Okay, so that's what we decided ahead of time is different between our two groups. That would be the environmental condition. So that's the independent variable. Okay, so think about what is the dependent and independent variable in the monarch butterfly experiment. So if we go back, we have the parasite infected and uninfected butterflies. Okay, so that's one independent variable. And then we also have the plants, right? So one type of plant is an anti-parasite chemical that has a high level, and then one is at a low level. So there's actually two, in this case, two independent variables. And then the dependent variable in this case is the number of eggs laid. So we're counting how many eggs are laid on each of the plants by each of the types of butterflies. Okay, and then finally, the other type of variables that we're interested in are the controlled variables. So controlled variables are variables that we keep constant among all treatment groups during the experiment. Okay, so these are something that all the treatment groups have that are the same. Okay, so for example, if we go back, we are using the same pots. We're using the same soil. We're using the same amount of water to water these plants. We're using the same types of plants. Okay, the only difference is the whatever type of chemicals. Okay, we're growing them in the same conditions. All right, so those are examples of controlled variables. Okay, so those are examples of the controlled variables in this experiment. So they are kept constant across all treatment groups during the experiment. Okay, and it's important that we do have controlled variables because we don't want to add any additional variables that can affect the results. So we don't want to have, for example, different amounts of water given to the plants, because then we don't know if maybe the water is affecting which plants the monarch butterflies are laying eggs on. Okay, so we have to make sure that everything's controlled for. All right, so in the next part, in Lab Study B, so we have choosing or designing the procedure. So what is a procedure? It's just the stepwise method. So throughout the course, you're going to be following different procedures. Though this is an online course, there will be times when you'll actually be performing some experiments in your home environment. So you'll, be familiar with procedures. If you've ever made a recipe at home, you've followed a procedure, right? So from start to finish. So it's the same kind of thing. The level of treatment, that's the value set for the independent variable. So you might have, in this case, we go back, our level of treatment are, the two types of plants, as well as, the two types of butterflies. So those are levels of treatment. Replication, so that's the number of times the procedure is repeated. So in this case, they did the experiment twice. And then finally, the control is a treatment group that does not receive the treatment. So sometimes there's a control group as a group for comparison. So in this case, we don't really have a control group. So we don't really have a group that we don't have, that we don't give the treatment to. So we have a treatment for all of them. But for example, if we were measuring, I don't know, if fertilizer has an effect on plants, we might have, different amounts of fertilizer given, to different plants, but then we might have a control group where the plant is given no fertilizer. So that would be the control group because it doesn't receive the treatment, the fertilizer. And that way it's going to be the comparison group. So if the group that didn't receive the treatment has the same result as the group that did receive the treatment, then we know that there was no correlation, okay? Or there's no, the results are not valid. So the control group is good for that reason, so that we can compare it. All right. In continuing on in exercise 1.2, next step, now that we have, if we go with our flow of information here, we've already learned about, observations and questions. You've practiced making hypotheses and designing an experiment. So the next step is actually to make a prediction. So predictions are a little different from hypotheses. So a hypothesis is an answer to the question. So that's usually, again, it's testable and it's falsifiable. A prediction is predicting the outcome of the experiment, and it either supports or falsifies the hypothesis. So it's a little different from just the hypothesis in that you're predicting the results of the experiment. So in this case, you need to know what the experiment is, and so it's a little bit more specific, okay? And it's usually written in an if-then statement. So for example, going back to the fertilizer plant experiment, if I give the plant more fertilizer, then it will grow faster than the plants that don't get fertilizer. Okay, so I'm predicting the outcome of the experiment. So that's different than just a hypothesis where I could just say that fertilizer affects plant growth rate. Okay, so I'm not really predicting the outcome of the experiment. I'm just, making a statement that is falsifiable and testable. So predictions, they're in if-then statements and they're predicting the outcome of the experiment. So I know that if I do the experiment and my plants grow faster in the treatment group that received the most fertilizer, then my prediction was supported, and it supports the hypothesis, right? So you'll practice, examining predictions and so forth. So that basically concludes pages 1 through 10 of your lab manual. Pages 11 through 15 have you do like an experiment, but since, we're not doing that, it's basically, you could do it if you really wanted to. It has you take your heart rate, and then you do like exercise, and then you take your heart rate again and see if your heart rate changes and so forth. So but we don't actually need to do that for this class. So you can skip pages 11 through 15 from your lab manual. Okay, so that's the actual doing the experiment and collecting data part. Okay, so skipping ahead to page 16. So this gets into, now that we've collected data, what do we do with that data? Okay, so it's presenting and analyzing the results. Okay, so the first part is basically compiling data into tables. So we need to know how to set up a table correctly. So here's an example of a table from your lab manual. So you can see that the table includes a title, and then we have the treatment, and then we have the results here, okay? So, you can clearly see that we have, the treatment. I can totally see what the experiment is just based on this table. I don't even need to read the information here. So it says treatment. I know there were 2 treatments. There was a control group, and then there was a group with SO2. Okay, and then so there must have been exposed to nothing and SO2, and then they counted the seeds per pod and then the pods per plant. So they were basically growing these plants in the presence of sulfur dioxide versus just regular air. And so they, it looks like they averaged it out. Okay, so average of seeds per pod. So looks like they averaged it out in this case. So they, 24 replicates. So this doesn't include all of the data. It's just they took the raw data and then they averaged it, it looks like. So they compiled it into a table. And so another way that, once we have it in this form, it's kind of hard to interpret it as a table. So next we make a graph. Okay, so in a graph is a visual representation of the data. And we talked about how to construct a graph in our previous video. So we basically do either a line graph or a bar graph based on if it's continuous or discrete variables. So in this case, you can see that it's continuous here because we have years, and then we have the number of wolves and the number of moose. Okay, so this, since time is continuous, we have this time being continuous, we have a line graph. All right, and you can see in this case we have 2 lines, so they indicated the 2 lines with color, and then they labeled the lines as well. And you can also see that in this particular graph we have 2 y-axes because the axes or the variables were too different. The population sizes were too different. We're in the thousands with the moose population, and in the up to 50 in the wolf population. We couldn't put them on the same axes because it would be read too differently. So we have 2 separate y-axes here. So that's just a little different for you to interpret. And then over here on the right we have a bar graph, and in this case, this bar graph, we have age, which is continuous, but the variable, the other variable is discrete. So we have men, women, and both. So we can't really have an in between because we have a both. Okay, so in this case, we have discrete data, even though you can think of years, well, that's continuous, but our other variable here being gender, we have 3 categories, so we can't really have an in-between a men and a both, or a women and a both, okay? So there's 3 discrete categories here, so we have a bar graph, right? So you're going to play around with graphing data. You're actually going to graph this data, okay? So think about how you're going to graph it. So we have, in this case, we have our treatment, so that's an independent variable. But this case we have 2 dependent variables, so they measured 2 things. They measured the seeds per pod and the pods per plant. So you have basically 2 sets of data here. So you need to think about, is this going to be a line graph or a bar graph? And how are you going to do this? You have, you're going to need to graph this data and this data, okay? So think about how you're going to graph it. So you're going to graph it they have like a little graph for you, right? And then finally, once you've graphed it, in exercise 1.5, you're going to basically, think about what you see here. What relationships can you see between the variables based on the results? Would it support or falsify the hypothesis? And then also, usually once you've made your conclusions, you can kind of think about, what were any weaknesses that you found when you were doing your experiment? Like, what was something that maybe you notice when you're conducting your experiment that maybe went wrong or didn't go as expected, or maybe something that you could suggest to improve your experimental design. Okay, so usually these are questions that we ask ourselves once we've interpreted our data. All right. Okay, so as far as our lab 1, usually what happens at the end of our lab, we have a worksheet that we fill out, but we're not doing it this week. Instead, you're going to take a quiz. Okay, so this quiz is based on pages 1 through 10 and 16 through 21. So you're not actually turning in any pages from the lab manual. Instead, you're taking this quiz. Actually, let me show you where it is. So you're going to go into our Unit 1, and if you go down to Lab Number 1, Scientific Method Exercise Quiz. So this quiz is available until Sunday midnight, and you can take this as many times as you want, okay, until you get 100%. And it's unlimited time, unlimited attempts, okay? So do it until you get 100%. All right, so this quiz, again, it's based on pages 1 through 10 and 16 through 21 of your lab manual. I think the last couple pages are just questions, so that's why it just says to 19 on your lab manual, okay? It's just 5 questions, but you do need to kind of complete the lab first before you take the quiz. Okay, so if you just skip ahead to the quiz, it won't make sense to you. All right, if you have any questions, feel free to email me. Otherwise, good luck.